Hi, my name is Carly Leninger. I'm a research associate at Mariah Thompson's lab at the NYU Langone Department of Child and Adolescent Psychiatry. And today I'll be talking about associations between prenatal maternal cortisol and the developing human brain. From a broad perspective, a central goal of the research at our lab is to discover fundamental properties of early human large-scale network development. Not only are we concerned with the fundamental biology, but also the operations of early environment that act on emergent neural structures, and most critically, the relevance of this variation to future health and behavior throughout the lifespan. Specifically, I have an interest in understanding the early environmental factors impacting network development related to future psychopathology. We like to refer to this environmental embedding as programming for the human life course, suggesting that adaptive neural development begins in the womb. In order to understand more about these prenatal functional blueprints, our lab has begun research on fetal functional connectivity using novel MRI methods. These are some fixed lab images of our fetal scans. I think they are absolutely beautiful, but the real issue is what can we learn from fetal MRI? So early on, our lab began to think about how much overlap exists in fetal and adult overall network organization. Here in this adult matrix, the top 10% strongest connections across the entire brain are shown as blue dots. You can also look at this as being sorted by neighborhoods they reside in, a kind of fingerprint for who's talking to whom. If you were to perform the same analysis in a fetus, how much overlap would you expect? We found that the fetal and adult matrices showed more than 62% overlap, indicating that yes, these early fetal networks resemble those persisting into adulthood. With this knowledge, we wanted to know more about the development of these early networks. With a baseline understanding that the same human neural networks of adulthood begin developing as early as in utero, it begs the question, how do environment and brain interact before birth to influence a child's future? There are countless factors of the prenatal environment, but today I'd like to focus on a biological factor, hormones, specifically cortisol. So why cortisol? Well, it's well known that hormones play a critical role in the development of functional brain networks. Cortisol specifically is known for its involvement with stress and complex cognitive processes. It does so through the HPA axis. The HPA axis is the physiological system regulating responses to stressors in the environment. This complex system involves a hormonal cascade from the hypothalamus to the pituitary gland to the adrenal gland, resulting in the release of cortisol throughout the body. Though this is a complex system, cortisol is the most readily available way to measure HPA axis activity in the humans, and so it's the component that we pay the most attention to. Cortisol has many targets in the body and brain, namely in the limbic region, including the amygdala and hippocampus, which are brain regions that are important for processing and regulating emotions. These regions also have glucocorticoid receptors, which make them sensitive to circulating levels of cortisol in the brain. This is a complicated feedback system, but much of the existing research tends to focus on cortisol and the limbic systems. In animals, preclinical studies have demonstrated that as early as in utero, the effects of circulating cortisol and induced stress are reflected in the offspring brain. Prenatal stress in rats has been associated with dendritic atrophy of pyramidal neurons of the hippocampus of the offspring. Additionally, rodent studies have demonstrated increased anxious behavior and increased HPA axis activity in the offspring as well, indicating that prenatal cortisol exposure is implicated in the development of both brain and behavior. Human studies that build on this work show that prenatal cortisol exposure predicts infant cognitive ability and child brain development as well. In a study conducted by Alice Graham, the potential impact of prenatal cortisol levels was examined through neuroimaging and behavioral questionnaires. Infant MRI scans revealed significant associations between maternal cortisol concentrations during pregnancy and neonatal amygdala connectivity in a sex-dependent manner. Behavioral questionnaires 2 years later revealed significant behavioral results as well, with a significant positive association between maternal cortisol and internalizing behaviors in girls only. These data lead us to believe that cortisol does impact the development of specifically the amygdala network. However, even neonatal scans cannot control for postnatal variables and/or birth-related effects. This brings us back to the question at hand, which is, do prenatal maternal cortisol levels impact the human brain beginning in utero? Data evaluating this association during pregnancy are fundamentally lacking. We aim to address this gap by examining prenatal maternal cortisol levels and functional connectivity in the healthy human fetus. Our pregnancy cohort consists of 57 women from the Detroit area. It is important to note the demographics of this population, since this cohort consists of predominantly minority women from under-resourced communities. So 90% have no college degree, 74% make less than $30,000 a year, and 80% of our cohort is African American. That being said, in terms of perceived stress, this set of participants had a fairly typical score, with their average falling within the low to moderate stress range. As for the infants, we had more males than females, with an average gestational age at scan of 32 weeks. For our scans, moms were scanned during the second and third trimesters. Salivary cortisol was collected from participating mothers every 30 minutes throughout the scan visit. 12 to 24 minutes of fetal functional resting state scans were obtained. Of course, fetal imaging comes with its own set of challenges, primarily high motion. However, having worked with these data for some time now, we understand that the fetus alternates between periods of high movement and quiescence. So the data I'll share with you today is only from those periods where the fetus remains still. But that's not the only problem. Additionally, there's no single orientation and a lack of automatic segmentation processes. And of course, the fetus is encased in independently moving maternal compartment. But despite all these challenges, we believe that scans are critically important to understanding the fundamentals of early human brain network development. So after pre-processing, we used the CON toolbox to generate maps of fetal limbic network resting state functional connectivity from bilateral amygdala seeds. We used a linear regression to evaluate how variation in cortisol relates to amygdala neural connectivity, controlling for gestational age at scan. Based on extensive literature, we focused our analyses on connectivity of the amygdala to key limbic structures, namely two prefrontal subregions, the anterior cingulate cortex, and bilateral anterior insula. Here, I'm showing you the results of that analysis. What you're seeing are regions within the fetal limbic network that show differential connectivity with variation in maternal cortisol. You'll notice significant clusters within our 4 anterior cortical midline structure ROIs, 2 in the ACC and 1 in each in the dorsal and the ventral PFC, and 1 in the anterior insula. Notably, the direction of effects was rather consistent. In 4 out of 5 areas, connectivity was increased in fetuses and mothers with higher cortisol levels, as seen in red. And the exception was this region in the ACC, where connectivity was actually reduced in fetuses of mothers with higher cortisol. Now, if we zoom out and look at total brain amygdala connectivity, we see that it is not just a story of high cortisol equals increased connectivity. Instead, increased cortisol produced a mixture of relationships across the entire brain. So it appears that the increased connectivity observed was potentially specific to limbic substructures. So to summarize, variation in fetal resting state connectivity suggests that effects of maternal cortisol levels on the brain may originate during the fetal period. These data support the idea that maternal biological factors can determine the optimal functional brain network development in utero. We also found that increased cortisol was associated with increased amygdala connectivity within limbic substructures. So overall, we've confirmed that cortisol exposure during pregnancy may alter development of major fetal brain networks, specifically areas including those later involved in cognitive emotional processes. There are, however, limitations worth mentioning. First of all, the evaluation of sex differences. Existing literature trends towards sex differences in outcome data, and we've yet to uncover those. More importantly, though, we've yet to evaluate the potential connections to behavioral outcomes. So we've looked at the associations between cortisol and brain development, but we haven't looked at brain to child behavior, nor have we examined direct paths from cortisol to child behavior. What role does this programming of biological risk play in future cognition and behavior, if at all? We know that altered limbic circuitry has been examined in translational and clinical research as having involvement with stress-related disorders and internalizing and externalizing behaviors. And we actually have relevant child behavioral data at ages 3 and 5. So the next step is connecting those dots to evaluate prenatal biological risk. However, with this understanding of fetal programming, it's important to remember that with plasticity, there's always opportunity. There are plenty of points for prevention and intervention throughout development to undo the impacts of biological prenatal environments. The first time point being the prenatal period, where things like social support, mental health resources, and adequate sleep and healthy diet can help downregulate the HPA axis. There's also postnatal intervention points where things like responsive caregiving, secure attachment, and enriched environment can encourage healthy brain network development. There's also adolescence, which is my personal favorite and a point of great interest for me, specifically because of that plastic opportunity that I mentioned before. Recent evidence characterizes adolescence as a potential critical period for higher order cognition. In particular, recent work shows that rats prenatally exposed to stress and stress hormones show improved behavior and HPA axis functioning due to environmental enrichment during periadolescence. So it seems like there is a potential for HPA axis recalibration and consequently potential limbic network restructuring. That's something that I'm really interested in. Overall, the data from these challenging fetal brain studies provides us with fundamental neurobiology that may hold the key to understanding human brain network development. A big thank you to all of our moms and families in Detroit. And also for me, a very big thank you to my mentors, Mariah Thompson and Michelle Van Tighem. Also shout out to Claudia Espinoza Heredia for her fantastic fetal processing, and a big thanks to our lab site at Wayne State for the data collection. Additionally, I would like to point out that I'm applying to grad school for a PhD in clinical psychology in the fall 2021 cycle, so please keep an eye out and keep in touch. My contact info is there. Thank you so much.